Biotherapeutics, including antibodies, have significantly improved global health. The key to the success of these drugs include its efficacy, manufacturability, and safety. However, during manufacturing conditions, it often destabilizes during the production processes. This can lead to it undergoing a phenomenon known as protein aggregation. That's where these proteins clump up and form these clusters and results in a loss of efficacy and can cause serious safety issues for patients. Despite the potential of over 700 monoclonal antibodies currently in clinical trial, the overall drug development process is still very costly and very complex due to all these instability requirements. To solve these challenges, my team and I leveraged a blend of experimental capabilities, theoretical modeling, and AI models to predict when antibody aggregation would happen and thus come up with ways to prevent it from forming. With these methods, we aim to rationally design biologics and formulations that can preserve their stability properties. This will help to cut down the time and cost of the overall development process. By unlocking the secrets to antibody aggregation, we hope to pave the way to the development of affordable treatments that are more reliable, safer, and importantly, more accessible. I'm Chia, a senior scientist at the Bioprocessing Technology Institute. My work focuses on the rational characterization and design of biologics for the developability properties so that we can translate the science to develop treatments for human diseases. If you want to know more about ASTAR science and technology, subscribe to our channels.